Good morning to all. The topic which I am going to deal today is the fourth species of comes under Chlorophyceae, Oedogonium, which comes under the family Oedogoniaceae and genus Oedogonium. Oedogonium is just like Cara. It is a multicellular macroscopic algae, which is a freshwater algae, and it is usually the plant body is unbranched. The filamentous and differentiated into apex and base. The peculiarity is the chloroplast is a reticulate network type of chloroplast is present in Oedogonium, and the second important point is presence of cap cells on dividing cells, which I will explain. And asexual reproduction takes place by zoospores, but the peculiarity is it is a multiflagellate zoospores, and sexual reproduction is of advanced oogamous type. Female gamete is known as Oogonium and male gamete is known as Androsporangium. And there are two types of plants based on the size of male filaments. They are Macrandrous type and Nanandrous type, which we will going to discuss in upcoming slides. So, as I already mentioned, the plant body is green, multicellular, unbranched, filamentous, and cells are arranged in a uniseriate row, and it is differentiated into a basal cell having holdfast and apical cell showing growth and middle cells. Basal cell is the lowermost cell. Usually it is colorless and performs the function of fixation and generally termed as holdfast. Whereas apical cell is the topmost cell of the filament, and cells is usually rounded towards apical cell and green in color. Whereas middle cell has some peculiar features. It's a presence of cap or cap cells. Okay, and the cell with cap is known as cap cell, and the number of caps on a cell indicates the number of time that particular cell divides. You can see, I will enlarge. You can see certain number of caps. See. There is 1, 2 cap, here there is 1, 2, 3 caps, sorry, and from this figure you can clearly see there is 1, 2, 3 here, 1, 2, 3, 4 cap cells are present, that is 4 times that particular cells divide. So, the intercalary cells are usually rectangular or cylindrical in shapes, and cells are surrounded by cell wall. The cell wall is also of peculiar nature. It is not cellulosic. It is not only cellulosic and pectinaceous. It is consisting of an outer chitin, which is usually found in exoskeleton of crustaceans. And just interior to the wall, cell membrane is present, which encloses protoplast, and all the cell membranes are cell organelles are present: chloroplast, pyrenoids, a well defined nucleus, and reticulate chloroplast. Cells contain vacuoles also. Chloroplast is present, which is large and reticulate. And why this cap cell is formed in every dividing cell is due to the peculiarity in cell division. All cells except apical and basal cells are capable of division through cell division. Okay. So, during cell division, what happened is, first a ring of hemicellulars appears on the lateral wall, and the ring slowly becomes thicker and wider. Next, what happened is nuclear division and cytoplasmic cleavage occurs, and cytokinesis results in a flowing septum, and cell wall then later ruptures at the region of the ring, and ring extends vertically. This is what happens. You can see the first nucleus just migrate towards upper portion, and a ring of hemicellulose has been deposited in the inner cell wall, and the ring extends, and you can see at the same time microtubules are, because cytokinesis starts between the two nucleus, and what happened is the cell wall break, the outer cell wall break, and when outer cell wall break, you can see that the inner pectinaceous membrane is elastic, so it extends in size. ആ സെൽ വാൾ ഔട്ടർ സെൽ വാൾ അങ്ങ് ബ്രേക്ക് ചെയ്യുമ്പോൾ എന്താ സംഭവിക്കുന്നതെന്ന് വെച്ചാൽ ഇലാസ്റ്റിക് നേച്ചർ കൊണ്ട് സെൽ വാൾ അങ്ങ് എക്സ്റ്റെൻഡ് ചെയ്യും. എക്സ്റ്റെൻഡ് ചെയ്യുമ്പോൾ നിങ്ങൾ കണ്ടോ, ആ റപ്ചറായ പോർഷൻ മുകളിൽ ഒരു റിങ് പോലെ ഡെപ്പോസിറ്റ് ചെയ്യും. ആ റപ്ചർ ചെയ്തത് ഔട്ടർ സെൽ വാളാണ്, അത് റപ്ചർ ചെയ്ത് മുകളിൽ ഒരു റിങ് പോലെ നിൽക്കും. ഇതിങ്ങനെ നല്ലപോലെ എക്സ്റ്റെൻഡ് ചെയ്ത് ഒരു സെല്ലിൻ്റെ സൈസിൽ എക്സ്റ്റെൻഡ് ചെയ്യും. എക്സ്റ്റെൻഡ് ചെയ്തതിന് ശേഷം സെപ്റ്റം കംപ്ലീറ്റ് ആയിട്ട് 2 സെല്ലിനെ ഡിവൈഡ് ചെയ്യും. രണ്ട് സെല്ലിനെ ഡിവൈഡ് ചെയ്തതിന് ശേഷം മുകളിലുള്ള സെല്ല് എന്താ സംഭവിക്കുക, ഔട്ടർ സെൽ വാൾ അങ്ങ് ലേ ഡൗൺ ചെയ്യും. ഔട്ടർ സെൽ വാൾ വിച്ച് ഇസ് കൈറ്റിൻ, ഔട്ടർ സെൽ വാൾ അങ്ങ് ലേ ഡൗൺ ചെയ്ത് കഴിഞ്ഞാൽ രണ്ട് ടൈപ്പ് സെല്ല് ഫോം ചെയ്യും. പക്ഷേ രണ്ട് ടൈപ്പ് സെല്ല് ഫോം ചെയ്യുമ്പോൾ പെക്യൂലിയറിറ്റി കണ്ടോ, ഇവിടെ ഒരൊറ്റ ക്യാപ് സെൽസേ ഉണ്ടായിരുന്നുള്ളൂ, ആ സ്ഥാനത്ത് ഇവിടെ എത്രയായി, 2 നമ്പർ ഓഫ് ക്യാപ് സെൽസ് ഉണ്ടായി. അപ്പോൾ ക്യാപ് സെൽസ് എന്ന് പറഞ്ഞാൽ വേറൊന്നുമല്ല, സെല്ല് റപ്ചർ ചെയ്ത് കഴിഞ്ഞിട്ട് അതിൻ്റെ ബാക്കി പോർഷൻ മുകളിൽ ഹാങ് ചെയ്തൊരു ക്യാപ്പായിട്ട് കിടക്കുന്നതാണ് ക്യാപ് സെൽ എന്ന് പറയും. ആൻഡ് ദ സെപ്റ്റം ദെൻ ജോയിൻസ് ദ പാരന്റ് സെൽ വാൾ ആൻഡ് പ്രോക്സിമൽ എൻഡ് ഓഫ് ദ റിംഗ് ആൻഡ് ഡിസ്റ്റൽ പോർഷൻ, അതായത് പൊട്ടി മുകളിലോട്ട് പോയ പോർഷൻ ബ്രോക്കൺ പാരന്റ് സെൽ പെർസിസ്റ്റ് ആസ് എ ക്യാപ്പ് അറ്റ് ദ ഡിസ്റ്റൽ എൻഡ് ഓഫ് ദ ഡോട്ടർ സെൽ. അപ്പോൾ ഇതാണ് സെൽ ഡിവിഷൻ. ഒറിജിനലി ന്യൂക്ലിയസ് രണ്ടായിട്ട് ഡിവൈഡ് ചെയ്യും. ഡിവൈഡ് ചെയ്യുമ്പോൾ തന്നെ ഇന്നർ സെൽ വാളിൽ നിന്ന് ഹെമി സെല്ലുലോസ് റിംഗ് മെറ്റീരിയൽ ഡെപ്പോസിറ്റ് ചെയ്യും. ആ റിംഗ് എക്സ്റ്റെൻഡ് ചെയ്ത് ഡീപ്പൻ ആവുമ്പോൾ ആ ഒരു പ്രഷർ കാരണം ഔട്ടർ സെൽ വാൾ ബ്രേക്ക് ചെയ്യുമ്പോൾ ഇന്നർ സെൽ വാൾ അങ്ങ് എക്സ്റ്റെൻഡ് ചെയ്ത് ഒരു ഒറിജിനൽ സെല്ലിൻ്റെ സൈസിലെത്തും. പക്ഷേ ആ പൊട്ടിപ്പോയ പാരന്റ് സെൽ വാൾ ഒരു ക്യാപ്പായിട്ട് മുകളിൽ റീറ്റെയിൻ ചെയ്യും. അതിനുശേഷം ഔട്ടർ സെൽ വാൾ ഡോട്ടർ സെല്ലിൽ ഫോം ചെയ്യും, സെപ്റ്റം കംപ്ലീറ്റ് ചെയ്യും, 2 types of cells are formed. Then upon cell division in Oedogonium is very important. Then reproduction, coming to reproduction, by 3 means Oedogonium reproduces: by vegetative, asexual and sexual. Vegetative by fragmentation and achainate. Fragmentation നമുക്ക് എല്ലാവർക്കും അറിയാം, due to breakage of the filament or dying of intercalary cells or intercalary sporangia, the fragments break and each fragments give rise to new filament. Achainate, during unfavorable condition, entire protoplasm become rounded off, become thick walled, reddish brown and oval structure called achainate. And achainate, during return of favorable condition, they germinate to develop a new filament. Then coming to asexual reproduction is also peculiar by the formation of a single large multiflagellate ovoid spherical zoospores from a single zoosporangium. And during favorable condition, this zoospores formation begins in the cap cells and entire protoplasm of zoosporangium rounded off. Nucleus and what happens is near nucleus, what happens is the protoplast entire become rounded and its nucleus moves at one end. And near the nucleus, a semicellular colorless hyaline portion develops, and just below the hyaline area, a ring of blepharoplast granules develop. And from each blepharoplast granule, single flagellum developed. Thus, a crown of flagellum is present around the colorless semicircular area, and the wall of zoosporangium breaks near the cap region, and the neighboring cell bend on one side to make way of liberation of the zoospores. ഈ ഫിഗറിൽ നിന്ന് നിങ്ങൾക്ക് കാണാം, എങ്ങനെയാണ് എൻ്റെ സൂസ്പൊറാഞ്ചിയം റൗണ്ടഡ് ഓഫ് ചെയ്യും, ന്യൂക്ലിയസ് ടിപ്പിലേക്ക് വരും, ടിപ്പ് പോർഷൻ മാത്രം കളർലെസ് ആവും. ആ കളർലെസ് ആയിട്ടുള്ള ന്യൂക്ലിയസ് ഉള്ള പോർഷനിൽ ബ്ലെഫാരോപ്ലാസ്റ്റ് ഡെവലപ്പ് ചെയ്യും. ബ്ലെഫാരോപ്ലാസ്റ്റ് ഉള്ളിടത്തെല്ലാം കുറേ ബ്ലെഫാരോപ്ലാസ്റ്റ് ക്രൗൺ പോലെ ചുറ്റും ഡെവലപ്പ് ചെയ്യും. അതിൽ നിന്നെല്ലാം ഈച്ച് ഫ്ലജല്ല റൈസ് ചെയ്യും. അങ്ങനെയാണ് മൾട്ടി ഫ്ലജലേറ്റഡ് സിംഗിൾ സൂസ്പോസ് ആവുന്നത്. അത് റിലീസ് ചെയ്യുമ്പോൾ സൂസ്പൊറാഞ്ചിയം ബെൻഡ് ചെയ്ത് ബ്രേക്ക് ചെയ്ത് സൂസ്പോസിനെ ലിബറേറ്റ് ചെയ്യും. Then during germination, the zoospores swim for 1 hour or more, and then the lower hyaline portion becomes separated by the cell wall, which forms the holdfast. Since holdfast is colorless, hyaline portion in the holdfast develop ചെയ്യും. അപ്പോൾ holdfast ഉള്ള റീജിയൻ വെച്ചിട്ടായിരിക്കും ഇത് സബ്സ്ട്രാറ്റത്തിൽ എന്ത് ചെയ്യുന്നത്, സബ്സ്ട്രാറ്റത്തിൽ ബൈൻഡ് ചെയ്യുന്നത്. എന്നിട്ട് through the subsequent division and redivision of the single plane, new filament is produced. Then coming to sexual reproduction. Sexual reproduction is advanced oogamous. Male gametes are known as androsporangium, which are produced in Androsporangium, and female gamete or egg is produced in Oedogonium. Male and female gametes differ both morphologically as well as physiologically. Only one egg is produced in each Oedogonium and 2 androsporangiums from each Androsporangium. ഇത് കൂടാതെ സെക്ഷൽ റീപ്രൊഡക്ഷനിൽ യു വിൽ എൻകൗണ്ടർ അനദർ ടൈപ്പ് ഓഫ് സ്ട്രക്ചർ നോൺ ആസ് ആൻഡ്രോസ്പോർ, വിച്ച് ഇസ് എ സ്പോർ, നോട്ട് എ സെക്ഷൽ വൺ, വിച്ച് ഇസ് എ സ്പോർ, ബട്ട് ഇറ്റ് ഈസ് നീഡഡ് ഫോർ സെക്ഷൽ റീപ്രൊഡക്ഷൻ ആൻഡ് ഇറ്റ് ഈസ് പ്രൊഡ്യൂസ്ഡ് ബൈ സിംഗിൾ ഇൻ ഈച്ച് ആൻഡ്രോസ്പൊറാഞ്ചിയം. ഓക്കെ, നമുക്കിനി ഓരോന്നായിട്ട് എക്സ്പ്ലെയിൻ ചെയ്ത് നോക്കാം. ഫസ്റ്റ് വൺ, based on the size of antheridial filament, the Oogonia are of 2 types, Macrandrous type and Nanandrous type, which is very important. Usually questions will be asked from Macrandrus and Nanandrus portion. First one is Macrandrus type. In this type, the Androsporangium develops as a normal size filament. But Macrandrus type are of two types, monoecious as well as dioecious. Monoecious means both Androsporangia and Oogonia are developed in a single filament, whereas dioecious means both Androsporangia and Oogonia are born on different filament. Okay. Whereas in Nanandrus type, what makes it different is they are always dioecious. That is, Androsporangia and Oogonia are always developed on different filament. They are of not two type, they are only of single type. They are always dioecious. In this type, Nanandria developed as a very small filament. It is not morphologically similar to the original vegetative filament. It is developed in a very small filament known as dwarf male or Nanandrium. Here initially what happens is Androsporangia are formed, and Androsporangia produced androspores. Androspores are liberated from the vegetative filament. Androspores swim for some times in water and they germinate on the oogonial wall or supporting cell, which I will explain, and forms a short male filament known as Nanandrium. And towards the apical region, the dwarf male filament cuts off to form antheridial mother cells, and from the antheridial mother cell, androspores are formed and they fuse with oogonium. Okay, so there is an indirect fertilization in Nanandrium with the formation of androspores. Okay. Whereas Nanandria species, as I mentioned, Macrandrus type are of two type, dioecious and monoecious, but Nanandrian type is always dioecious, but they are of two type, Gynadrosporus and Oedogonium. Gynadrosporus means Androsporangia and Oogonia are born on the same filament, not Androsporangium and Oogonium, Androsporangium, which produce androspores, and Oogonium are produced on the same filament, whereas in Oedogonium, the Androsporangium and Oogonium are born on different filament. Okay. So, we will explain first the sexual reproduction is Macrandrus type. Okay. Here there is no morphologically dissimilarization. Both Androsporangia and Archegonia are born as the same filament length. First we will discuss on Androsporangium. Any cap cell, any young cell which undergo division can function as androidal mother cell. It first transversely divides into an upper smaller androidium and a lower larger sister cell, and the sister cell then undergo repeated division to form 2 to 40 rectangular uninucleate androidia, and the nucleus of androidium undergo mitotic division and forms 2 nuclei. Each nucleus becomes surrounded by cytoplasm and metamorphosed into androzoids. So, any capsule can function as androidal mother cell. They transversely divides into an upper smaller androidium and lower larger sister cell. Sister cell again undergo 2 to 40 divisions to form uniceriate androidia, and each androidia undergo mitotic division to form 2 nucleus, and each nucleus becomes surrounded and metamorphosed to become androzoids. Thus, 2 androzoids are produced from each androidium. Androzoids are uninucleate, unicellular, multiflagellate and yellowish in color. They are morphologically similar to zoospores and androspores but smaller in size. And then when mature, the androzoids are liberated, and just like zoospores, they swim in water for some times. Then coming to Oogonium, any cap cells can function as oogonial mother cell. It also divides transversely to form an upper oogonium and a lower supporting cell or suffultatory cells. The lower cell may again undergo similar divisions to form 2 or more oogonia with a lower supporting cell. With maturity, the oogonium become globose, which contain only a single egg, and a receptive spot is present on one side of the egg. Before fertilization, a transverse slit or pore develops on the oogonial wall, through which androzoids takes its entry. Okay, so Macrandrus species is very simple. Whether both the androidium and archegonium or oogonia are developed from the same filament, or both male and female filaments are present in dioecious species. Then in Nanandrus species, which is little bit complicated, there are 3 different structures: Androsporangium, Oogonium and Androidium. Okay. First one is Androsporangium. Androsporangium, the development is just like Antheridia. Nucleus of Androsporangium does not divide, but the only difference is the entire protoplasm. Just like the androspore mother cell divide transversely into upper and lower cell, the lower cell again divide transversely to form 2 to 40 Androsporangium, and by Androsporangium, the entire nucleus of Androsporangium round off and forms single androspore. Not like anthrosooids. Androspores are unicellular, multiflagellate and larger than anthrosooids. They are liberated by the rupture of wall of Androsporangium. During liberation, each androspore remains coated in a mucilage for some times and then become free to swim in water. Here in Gynadrosporus, you should remember that from the same filament producing Oedogonium, Androsporangium is developed. This is how androspores are looking like. Androsporangium, empty androsporangia liberating androspores. You see the androspores comes and settle in the suffultatory cell or supporting cell of Oedogonia, and they germinate to form a short filament. See, a short male filament known as Nanandrium or dwarf males. Nanandrium, which they can germinate in oogonial wall or just the cell below the oogonium, that is a supporting cell. And after swimming, that what I already mentioned, after swimming for some time, it get attached in supporting cell or oogonial wall. Then a wall developed around the androspore. Androspore elongates and divides. Nucleus of each Antheridium divides mitotically, just like in Macroandrous species. Each Antheridium divides mitotically to form 2 nuclei. Each nuclei is rounded off with some cytoplasm and metamorphoses into single anthrosooids. Thus, 2 anthrosooids are formed in each Antheridium. They are liberated in a similar way as in Macroandrous species. They swim for some time and come in contact with the receptive slit of Oogonia, and they enter inside the Oogonium and fertilizes the egg. Okay. So, Oogonium, the structure of development of Oogonium are same in both Nanandrous and Macroandrous species. So, after fertilization during fertilization only one anthrosooid enters through the opening of Oogonial wall and fertilizes the egg, resulting in the formation of a diploid zygote. The zygote during further development, they secrete say 2 to 3 layered outer wall and form the oospore. Later the outermost wall may become ornamental, and zygote generally undergo period of rest and then becomes brown in color, and they germinate during return of favorable condition. And since this is a haplontic species, the nucleus undergo meiosis and forms 4 haploid nuclei. Nuclei accumulate some cytoplasm and forms 4 daughter protoplasts. They liberate by rupturing the oosporial wall. They develop flagella and they function as meiospores. That is, after meiosis, zoospores are developed. So, they are known as meiospores. Initially, they remain inside a vesicle, then disintegrate, and the zoospore get free into the environment. After swimming, they withdraw their flagella and germinate into new haploid Oedogonium filament like zoospores, that is with its hyaline portion attaching to the substratum, and the hyaline portion forms a holdfast and the rest forming the vegetative filament. Okay. So, this is what about the life cycle of Oedogonium. You should always remember that in Oedogonium 2 types of species are present, Macroandrous species and Nanandrous species. Macroandrous, just like all other algae, both male and female filaments are morphologically similar, and male filament from the male when it is in a homoecious species, both male and female produced in the same filament. Whether it is when it is a dioecious species, male filament is also present and a separate female filament is present. But in Nanandrous species, you should encounter an intermediate stage known as Androspores, which is present in same filament producing Oogonia. This is known as Gynadrosporus, or Androsporangium can be produced on a separate filament known as Oedogandrosporus. Whether separate or same, development is similar. Androsporangium produce androspores liberated, and they germinated in the supporting cell or suffultatory cell of Oogonial wall, and they germinate to form a short filament, a dwarf male filament known as Nanandrium, but it is parasitic to the female plant. It is not a separate filament, and the Nanandrium give rise to Androzoid. Androzoid liberated and they fuse with the same Oedogonium to form the zygote. Zygote, after undergoing a period of rest, they undergo meiosis to form 4 haploid protoplast. And the protoplast metamorphosed to form meiozospores. They liberated and germinated to form the haploid oogonial cell. The 2 of the filament can form, 2 of the protoplast can germinate as male filament or 2 as female. These are the life cycle of Macrandus species. Just go through it. I am not explaining. This is the life cycle of Macrandus dioecious species. This is the life cycle of Nanandrius, that is Gynadrosporus species, whether male and female, whether Androsporangium and Oedogonium is, Androsporangium and Oedogonium is present in the same filament. And this is the life cycle of Nanandrius Androsporus species. You can see there is a female separately producing and there is a filament producing a Androsporangium. Okay. So, these are all about Oedogonium. It is very peculiar in having cap cells, chitinaceous cell wall, no algae having chitin, very few algae. I think in your syllabus only Oedogonium having a chitinaceous outer cell wall, and the presence of cap cell and the presence of Macrandus and Nanandrius type of species makes Oedogonium a very interesting species to study. So, study well, go through it and write down, prepare notes and be prepared for an exam on next class. Thank you.